Okay, we're going to continue our talk now, talking about more structures that are within the cell. And we've got some pictured here on this slide. The leucoplast is an organelle that's primary function is to store starches and oils. In contrast to the leucoplast, we have something known as a chemoplast, or a chromoplast, would be a better way to state it. These are organelles that contain pigments, thus the term chromo, which means color. And one example of a chromoplast is a chloroplast. A chloroplast has chlorophyll, which is necessary for photosynthesis. So the chloroplast is where the photosynthesis occurs, and it is a structure that is necessary for that function. As I said before, in the plant cells, we also have something known as a central vacuole. I pointed that out on a different diagram that we were looking at the last part. This is a specialized central sac structure that's seen only in plant cells, and this is filled with a solution that contains many solutes, not the least of which is glucose. Now, there are processes other than secretion and excretion and absorption that are ongoing. And you remember I emphasized the fact that many of those processes were soluble substances or dissolved substances. Well, we're going to look at some processes now which require the uptake of non-dissolved substances. And the first of these is shown on the left here. It's called phagocytosis. This is phagocytosis. This is how phagocytosis works. And phagocytosis occurs when a cell engulfs something that is not dissolved. And what you can see here is a large particle. This could be a bacterium. It could be just a large particle of something on the outside of the cell, and the cell decides that it wants it, and it takes it in this way. Now, this is very similar. What's called receptor-mediated endocytosis is very similar, except for the fact that it identifies what it wants to take in by these receptors that are in this vesicle. Once you've taken in a substance, it is placed in a round structure that's called a phagocytic vacuole. And within the phagocytic vacuole is now the particle that's been taken up. Now, sometimes other vacuoles come and dump acid or molecular acid in here, and this becomes like a lysosome. And you see here that this also has a somewhat thick membrane in order to prevent the escape of this material from this vacuole. Now, the other process is called penocytosis. Now, penocytosis is the uptake of fluid as opposed to particles within the cell. And this results in what's known as a penocytic vesicle or penocytotic vesicle. And it takes in these fluid materials. And it's not the same as absorption because you don't form specific vacuoles with the fluid in it. Now, if you just look at this in reverse and say that this is a material that the cell wants to discharge and it goes up and expels it this way, then you have what's known as a secretion vesicle. And the only difference between a penocytotic vesicle and a secretion vesicle is with penocytosis, it's going into the cell, and with a secretion vesicle, it's going out of the cell. Now, this diagram shows I put a circle around what's called a Golgi apparatus, and that's this sac-like structure. It's been described variously as a stack of pancakes, okay? And the Golgi apparatus, the purpose of the Golgi apparatus is the storage of proteins and lipids. So once proteins are made here in the endoplasmic reticulum, they're frequently transported to the Golgi apparatus along with lipids. Then in this area, they're stored, and frequently the proteins are then modified in some way depending on what the cell needs. And so that's the purpose of the Golgi apparatus. The other structure I want to point out is this one here. And you'll notice that you have one that's sort of directed this way and one that's directed this way. And this is commonly the way the diagrams show this centriole. They'll show it as two organelles that are at somewhat right angles to each other. And the centriole has a lot of specific functions. One function is that it forms the basis for the cilia that come out of cells that have cilia. It also can form the basis for the flagellum that is formed. In most mammalian cells, it functions to form microtubules. And what will happen is, as it forms microtubules in the process of mitosis, the centriole will migrate to the poles of the cell, one side and the other, or the pole, and then there will be a separation. The microtubules, the purpose of the microtubules is to pull the chromosomes apart. So the microtubules are strands of protein that form tubes that are frequently used to move things around in the cell. And the centriole is important in mitosis and the active dividing of cells. Now when we move to the nucleus, this is a representation of the nucleus. The nucleus is where all the DNA is stored, okay? And the outside on the DNA, you'll have this structure which has very large pores in it. This outside structure is known as the nuclear membrane. And the nuclear membrane, because of the purpose of the nucleus, has to be very porous and allow for the passage of large molecules, particularly RNA. Because RNA passes in, or RNA is created in the nucleus and then passes out through the nuclear pores into the cytoplasm where ribosomes attach and make protein. And so this nuclear membrane surrounds the nucleus and protects it from a dissociation of the nucleus, but it does have fairly large pores that allow for transport of large molecules. Now, DNA is frequently formed in strands called chromatin, and this chromatin generally has associated with it not only DNA, which is the central aspect of it, but also RNA and proteins. And then within most mammalian and animal nuclei, you'll see these dark structures here and here. And this dark structure is called a nucleolus. And a nucleolus is where transcription occurs. And transcription is the process where DNA is used as a template to form RNA. And then the messenger RNA, which is used to make protein, then transfers outside the cell into the cytoplasm, and then translation occurs, and that's when the RNA is used to make protein. Now this is a kind of an interesting looking diagram, and what it's made to show is this real fine structure here, and those are called, this is called the cytoskeleton, excuse me. This is a network of fibers within the cell that essentially has two purposes. Well, really three purposes. First of all, it holds the cell together. Secondly, and it keeps the organelles oriented in a certain orientation. Secondly, it keeps the cell in its shape that it forms. So it's like a skeleton, thus the name cytoskeleton. And then thirdly, it aids in moving things around in the cell. The cytoskeleton is important in helping generate cytoplasmic streaming, which we talked about before. Now, the microfilaments are these fine thread-like proteins that are found within the cytoskeleton that are the primary components that make up the cytoskeleton. Now, also part of the cytoskeleton are filaments called intermediate filaments. The intermediate filaments attach microfilaments to each other. They're approximately twice as thick as the microfilaments, and so you have a network of thick and thin filaments throughout the cell that do the functions that I've mentioned previously. And so that ends the second part, and this week we only have two parts, I only have a total of about 25 minutes of talking, so you should have plenty of time to read what's in your book and do your homework. Thank you and have a nice day.